Hi, everybody. Thank you very much for coming. I know it's the last session and you are dying to go home or at least going for a nice to the Rio Grande. So we will not be long. So I would like to start this session. It's about antifungal resistance. And our first speaker is Damian Krysan. So please, Damian, come. He will be talking tell us about tolerance, hetero-resistance, and resistance. Thank you, Damian.
Well, thank you very much to John and the organizers for asking me to participate in this meeting. It's the first time I've actually been to mycology study group meeting, so it's been a pleasure to be here. I take this topic that was given to me with some trepidation. There are a lot of folks in the room, or at least somewhere in the area, that probably know more about some of this stuff than I do. But I've decided to take the title and change it just a little bit. And what we're going to focus on here is what we talk about when we talk about antifungal drug resistance. And let's see, what do I push? This one? The green. And so that title actually comes from a short story title from one of my favorite short story authors, named Raymond Carver. And he also had a quote at one point that says, "That's all we have, finally, the words. And we had better get the words right." And so what I'm going to try to do today is go over some of the words that we use. To begin with, I have, unfortunately, no conflicts of interest. And I'm not going to talk about any drugs that are off-label. And so what I am going to talk about is in the reverse order of which John suggested. I'm going to start with resistance, move to hetero-resistance, and then tolerance. Kind of going from the more defined to the less defined of these terms, in my opinion. We're going to talk about some formal definitions, some operational definitions, some examples that I hope that will prove illustrative, and a few of my thoughts on clinical implications for particularly hetero-resistance and tolerance. And so resistance, I think, is the one concept here that we can come to the most common ground on, although there is some ambiguities that can emerge. And so here we have basically a clinical microbiological definition. A strain, or an isolate, has a minimum inhibitory concentration determined in the laboratory under standard conditions, and that MIC is higher than a clinical cutoff. That's resistance, R, for us clinicians. What does that mean? It means that a patient that is infected with that particular isolate is less likely to respond to that therapy than one that is a patient who is infected with an isolate that is below that cutoff. That's pretty much all it means. This is really a binary usage. It is resistant or it's susceptible. Now, before anybody gets cranky, I realize that there is something in between called an I. Stands for intermediate, indeterminate, I don't know what to do, call ID. Yeah, but in general, this is R and S. There's no extent to that definition. So in other words, there's no more resistant or more susceptible, or less resistant or less susceptible. It is a cutoff. It's like saying you're more or less dead. All right, so that's fairly straightforward, clear-cut for the most part. To give you an idea of what a journal title would be that's using this particular definition of resistance would be the prevalence of fluconazole-resistant C. albicans isolates, pediatric patients treated with fluconazole in the US between 2000 and 2015. You will have, if you open that, or now you open the file and you look at it, and then the paper will describe MIC values that were generated using either CLSI or UCAST methodologies, and they will be compared to either clinical cutoffs or sometimes epidemiologic cutoffs. That's a pretty standard paper that we see. So that's using the very specific clinical microbiology definition of resistance. So, but that's not the only way we use this term, it turns out. There's a little bit more informal and idiosyncratic definitions. And these are widely used, particularly in the basic sciences, in some of our actually resistance mechanism literature. In this use of the term, it is really synonymous with susceptibility, more or less susceptible. So in this usage, the term has extent. You can have more resistance. People use the term, this is highly resistant, highly susceptible, exquisitely susceptible, dangerously resistant. All right, so you hear this, right? What it means is strain A is called more resistant when it has an MIC that is much greater than some reference you're starting with, usually a wild type. A lot of times you'll see spot dilution assays or some sort of growth assay, and the mutant you're studying has much higher MIC or much less more growth on a given concentration of drug. And so this is really associated with a phenotype as opposed to a clinical definition between resistance and susceptibility. And you'll read this in the literature quite a bit. All of us have come across it. An example of a journal title that is using this formulation of the term resistance would be your favorite gene 1 is required for virulence and antifungal drug resistance in Candida albicans. You go and look at this paper, and what you will learn about is the lab has knocked out their favorite gene in a standard laboratory strain, did a bunch of phenotyping, found that it grows poorer on fluconazole or caspofungin or something else than the starting SN250 or SC5314 parenteral strain. No drug-resistant strains are involved in this paper. So what you're describing is a change in susceptibility from a wild type, and it is not necessarily related to clinical microbiological definitions. All right. Other terms that we can talk about are acquired versus intrinsic resistance. And so acquired resistance is basically a drug is active against the majority of the isolates of that given species. And some isolates, however, are found that have an MIC that is much higher than the cutoff determined by the CLSI or UCAST. Perfect example of this was when Tom Schiller this morning was talking about Candida auris that developed echinocandin resistance on therapy. That is a strain that started off being susceptible to echinocandins and then later in the treatment course is now resistant. That is acquired resistance. Generally, that occurs because of one of a number of usually by now actually pretty well-defined molecular mechanisms that are determined by the chromosomal changes to those genes. So that's acquired resistance. Intrinsic resistance. This is where the majority of the isolates of a given species has an MIC that is so high that we cannot achieve drug levels to treat that patient with that drug. And so in this instance, we're using actually a clinical microbiological definition of the term resistance. And an example is Cryptococcus neoformans. Famously not susceptible under clinically meaningful concentrations to caspofungin or any other echinocandin. Now you can put 32 to 64 micrograms per mil into your culture of Cryptococcus and it will stop growing. But that is way, way higher than what you can achieve in a human. And so this is defined as intrinsic resistance. It doesn't mean the drug will never inhibit the growth of that organism. There are some ways to use this more idiosyncratically. We hear many folks say Candida glabrata is intrinsically resistant to fluconazole. It's actually not technically true. If you look through most of the epidemiological studies, based on our cutoffs, clinical cutoffs, most of the isolates of Candida glabrata actually are What is different is that cutoff is super high compared to other Candida species. It's around 32 micrograms per mil. If you try to treat Candida albicans patient who has an isolate that is at 32 micrograms per mil with fluconazole, you will most likely fail. So these are differences in sort of how we use this. And my goal here today is not to get you to become, sort of a grammar fascist about the use of these terms, but use them in a consistent way in your publications and your manuscripts and describe how you're using those terms. That will help, I think, with a lot of sometimes the confusion that can arise from the mixing of the more formal and the idiosyncratic uses of the terms resistance. All right, now we're going to go toward the less clear-cut, the more ambiguous. So, hetero-resistance and tolerance. Tolerance is the most recently developed term, and I'm going to talk about that last. Hetero-resistance probably was, at least in fungi, was originally described in Cryptococcus by Junwan Quansheng about 15 years ago. And what is different between these two terms and what we were just talking about is that this is a population characteristic. When you look at the MIC of a given isolate, you're looking at growth in a bulk culture, and you're looking at the majority outcome of that. And some folks started to look deeper into those cultures and were surprised to find that not every cell in that culture had the same susceptibility. Unfortunately, these terms have been used quite inconsistently, and their definitions are ambiguous, both in the antibacterial and antifungal literature. So, one of the reasons I think I'm up here is that Judy Berman, and I helped Judy Berman write an article a few years ago on trying to, sift our way through some of this. And this is a figure that comes from that article. And so if you look at the first line you come to as you go along, you come to a blue line. And that blue line is a susceptible species. And on the x-axis we have CFUs. So these are cultures, think of this as quantitative cultures being plated, and you're counting the number of organisms. And the first line, nice, beautiful dose-response curve, is a susceptible isolate. Now let's go to the purple line, the one farthest that way. Much bigger area under the curve, nice dose response, but that particular isolate is resistant because it takes way more drug to get down to very low concentrations of remaining colony-forming units. So that's easy. We just talked about that. The two in the middle represent hetero-resistance and tolerance. The one that still has a dose-response curve, the red one, is a hetero-resistant isolate, in which the MIC is changed by about at least 8-fold. The green line represents a tolerant strain. And we'll talk about those two one by one now. So, hetero-resistance, subpopulation, in other words, it is a fraction of the total inoculum that grows at a MIC 8-fold higher, okay? If you take that strain, pluck it off your plate, passage it in drug-free media, and you do the same experiment again, you get the same result. You will get, if you had 10% of the population grew at 8-fold MIC of the main population, it will have the same exact proportion growing the second time you did it. So this is a metastable change in the MIC of an isolate. In other words, it is, if you do not get a high MIC from that strain. So, I think a graph graphical representation of this is in order. So if we take isolate A, when you have 10 CFUs, and we plate it on drug X at MIC, and we end up with 2 isolates that survive at that concentration. 80% of the cells died or did not grow. You now subculture that population, 1A and 1B. Passage them on drug-free media, and now what you do is you replate on that high concentration of drug. And for a hetero-resistant isolate, what you get back is 10% again. Exactly the same. And then it turns out B is not a hetero-resistant isolate because when you plate that, all of them grow. That is a spontaneously generated resistant mutant, the sort of thing we look at when we're generating data, for preclinical data for a new drug. You want to know how quickly or how common is it for resistance to emerge, spontaneous resistance rate. If you could have a really high rate, not a great drug. Something like flucytosine, very high resistance rate. Okay, so that's the difference between resistance and heteroresistance. So, to reiterate this a bit, the way I like to think about it is heteroresistance is actually characteristic of the isolate. So if you take 25 isolates, you may have 10% of the strains are heteroresistant, 1% of the strains, 25%. It varies from strain to strain. Resistance itself is characteristic of the drug, not the strain. Because if you take a whole bunch of them, you will get usually, again, use 5-FC as an example. You take a bunch of strains of 5-FC, you get 20 to 30% resistance after X amount of time. And that's pretty consistent across different isolates. And these are generally due to chromosomal mutations, I said before. To date, heteroresistance is most commonly associated with aneuploidies. Duplications, triplications of chromosomes that contain things like ERG11, the target of fluconazole, or efflux pumps. All right, now we're down to the one that's probably least well defined and least well understood at this point. Tolerance. Tolerance is actually quite similar, if not identical, to another term that was used in particular with fluconazole susceptibility, and that was trailing. So they start with T. You can keep those two in mind, right? This is really what we used to call trailing. Drove everyone crazy about how to call an MIC for fluconazole for many years. It is basically slow growth at drug concentrations that are above the formal definition of the MIC. And so for fluconazole, for instance, you call your MIC as a clear-cut reduction in growth from the untreated control. Sometimes it's referred to in IC50. You don't use the same endpoint call when you're testing fluconazole as you do for an ichinocandin, which is based on a clear well. All right, so you notice there's growth out in those wells, maybe particularly a day later, you'll see a lot of growth in those MIC wells. And you can still see the same thing if you do, say, an E-strip test, and in the zone of clearance, first day you look at it, wow, that's great, nice clear definition of your MIC. Come put your plate back in the incubator, come back, and there's all this stuff growing clearly inside the original zone of clearance. And that's actually the stuff that Judy Berman got interested in. She wanted to know what that stuff was. And she developed this term called tolerance. And it's similar to heteroresistance in that if you take cells from inside the zone of clearance and outside the zone of clearance, passage them, put them back on drug, you get exactly the same pattern every single time. So this is a metastable, right? It is not due to chromosomal changes in or mutations. It is a characteristic of a given strain, and the amount of tolerance can vary from one to another. And there are now some criteria that she's generated to score that sort of tolerance, all right? And so one of the things that differentiates tolerance, well, here, let me give you an example of this. So this comes from a paper that was published in 2019, and it's a fantastic study from Tihan and Bishanek. Judy Berman was involved, Stone et al., where they took Cryptococcus directly from patients and LPs, plated them, and were looking at the fluconazole susceptibility patterns of those isolates. And one of the figures from this, figure 2, gives you a very nice demonstration of the difference between tolerance and heteroresistance on a plate. So we have a YPD plate, and next to that, I hope this is coming through. It's not coming through great for the audience. You see this really kind of fuzzy looking colonies in the background on the tolerance, and it's uniform across, and it's about 20% of the YPD determined CFUs. Then you go to a higher concentration at 64, and now you start to see what look like suppressor mutations to all of us who've done genetics in any fungi. Those are the heteroresistant colonies. If you take them and maintain them on fluconazole, they will grow at high concentrations. If you take them off of fluconazole and you replate them, you get that exact same pattern back. And the same thing with tolerance. And so, what are the clinical implications of this? And for tolerance, we really don't know. In the initial studies that evaluated the trailing effect in fluconazole, many studies were done with animal models that showed that there was really no measurable difference in outcome in experimental treatment of Candida albicans, in particular with fluconazole, in terms of whether you treated one with a lot of trailing or with no trailing. And so far that is really, we've done some experiments along those lines with high tolerance and low tolerance strains, and we can't see a difference in those models. There is an association that Judy's lab reported where there was slow clearance, so clinical response for some strains that had higher tolerance levels. That was not a prospective study. There's a lot more to do to firm up that sort of a correlation. Hetero-resistance, on the other hand, is probably the most stark example of how this sort of population response to drugs has clinical implication. And again, this is from Stone et al., where they have shown that the reason fluconazole monotherapy, or at least one of the reasons fluconazole monotherapy is so poor, is that heteroresistance emerges very rapidly, and essentially you end up with resistant isolates that are, or resistant Cryptococcus in the patient after only a short period of time. And just to show you some of that data, this is again from Stone et al. 2019. On that side, you have the EFA that we've seen quite a bit of lately for fluconazole by itself. If you add flucytosine to that, you go from a fairly flat EFA to one that at least has some slope. And if you look below, the monotherapy in the middle, that's fluconazole monotherapy, and it has the proportion of heteroresistant colonies that emerge on fluconazole monotherapy when you're doing the LPs over time. You can see the heteroresistant rate is much, much higher than when you add flucytosine, and you get essentially no heteroresistance. And so one of the things I'd also like you to sort of remember is that the starting CFUs per mil in Cryptococcus are extremely high, like 10 to the 6th, 10 to the 7th for some patients. That's an OD. That is an OD of cells in the CSF. So you have 10 to the 6th cells to 10 to the 7th per mil. You have 20 or 30, I don't know, a lot of CSF there. So you have a huge population of slow-growing organisms that are exposed to a static drug. That's how you generate hetero-resistance. You have a huge opportunity for those cells to, there's genetic diversity in there. You select for that hetero-resistant pair or population, and it sweeps the population in the patient unless you have something that gets in the way of that, like flucytosine. All right, so again, I said I don't think we're going to get rid of the idiosyncratic, nor necessarily should we get rid of the idiosyncratic use of resistance. But I implore you all to use it consistently and define it in the way that you're using it in the papers that you write. That will improve our communication, I think. Hetero-resistance and tolerance refer to variation in response to the drug of subpopulations. We get it in our head that everything's clonal. Well, by the time you have a single colony that you can see on your plate, you have 10 to the 7th cells that started from 1. You have heterogeneity, diversity in each colony. Just, we've heard of how many SNPs there are and the different variations we've heard about today. So remember that these things are not completely clonal and that there's variation within subpopulations. The clinical importance and the mechanisms of this are under investigation. It's still to be shown what the relevance to tolerance has, but certainly mechanisms of hetero-resistance and its importance in clinical failure for our patients is something that I think deserves a lot more study and will hopefully provide insights to how we can better design our therapies. In this case, the 5-FC flucytosine story really provides a nice rationalization why that's so much better than fluconazole alone. And here are some references that you can hopefully will be helpful to you. And I thank you for your attention and the opportunity to talk about it.